Well, thanks to Ashok and Eric for giving a great introduction to CLS. And that actually allows me to get right to the point of my, let's get away from that. So I was going to give a brief introduction to Cercospora, but my colleagues have done a fantastic job of covering that, so I won't dive into that too much. Importantly, though, keep in mind as we go through this presentation that Cercospora is a hemibiotrophic fungus, which is, again, a fancy way of saying it's got a symptomatic and an asymptomatic phase. And it is a polycyclic pathogen. So as Eric very nicely highlighted with his chart showing sort of the exponentially growing problem of Cercospora as the season progresses and new lesions develop, each spore that lands on a leaf you can think of as causing potentially a single lesion, likely more than a single lesion, and each spore then has the capacity to generate thousands of additional spores. And so really the best time of the year to get a handle on CLS is right away when that primary inoculum is coming into contact with your sugar beets, because every single successful infection past that will start to produce more and more lesions that you have to deal with. Now, in the work that Dr. Secor and my colleague Dr. Secor at NDSU Plant Pathology has been working with me to develop a more comprehensive understanding of the CLS disease cycle. And a ton of work had been done in the polycyclic phase of this interaction, which is this big giant circle here in the middle. But not a ton of work had been done in this pre-symptomatic phase to sort of map out the different steps of this infection mechanism. And so Dr. Secor and I sort of laid out a few questions we wanted to address, such as when do we see spores dispersing, what conditions lead to spore germination, and then knowing those two things, can we detect latent infection and get a handle on the timing of CLS's the true start to infection. And Dr. Secor did a fantastic job of getting a jump start on this even prior to me being hired in 2021, when he and his technician Viviana looked at the conditions that promote spore germination here. And they found a really interesting result that the presence of free water, so highlighted here in this yellow-orange box, the presence of free water had a much, much stronger effect on spore germination than high relative humidity. And this is important for us when we're talking about identifying the start of infection. Typically, we always sort of relied on high levels of humidity to indicate that we might have a risk of infection beginning. But now knowing that free water is significantly more important, we can start to model free water events that are typically high rain events, which this year we saw a ton of rain in May, June, and July. And also importantly, dew formation on the leaves for extended periods of time. And those water droplets then provide the necessary fuel for Cercospora spores to start germinating, and for that then hyphal tissue to find its way into a sugar beet leaf. Dr. Secor also narrowed down some other modeling parameters for us with the temperatures that you can see here that they tested from 10 degrees Celsius, so 50 degrees Fahrenheit, all the way up to 68 degrees Fahrenheit to measure the effect of increasing temperatures on spore germination. So as you can see here, temperatures increase, you get more spore germination. But they had plenty of spore germination at temperatures as low as 50 degrees. And then they did a nice job of testing how long spores need to be exposed to that free water by testing 2, 4, 8, 18, and 24 hours of exposure. And as you can see here again, you have a sharp increase in spore germination with longer exposures to free water. So armed with that knowledge as to the conditions that spore germination should be occurring at, we then leveraged our latent infection screening with our molecular tools to see if we could track down and identify when this infection actually begins. And we're using a multiplex qPCR assay that was originally, each of the markers that we run for detecting Cercospora and fungicide resistance mutations were originally developed in the Bolton lab at the USDA. And we've basically taken and applied those now to detecting latent infection. And the results of that in the Red River Valley have been really consistent over the last 4 years of this testing, where we see this sharp increase in the prevalence of fields that have latent CLS detected, typically in that latter half of June. And you can see that here, where from the 3rd week of June to the 4th week of June, we get a sharp rise in latent CLS detection. And in southern Minnesota, that actually happened, this big jump here is typically fueled by the samples from southern Minnesota. So you guys are seeing slightly earlier latent onset compared to more northern fields. One thing from this year that was atypical compared to the last 3 years, typically we see a higher prevalence of strobilurin-sensitive spores, and the latent infection typically for us starts with higher levels of strobe sensitivity. This year we had resistance show up in our assays much earlier than we've ever seen, and one of the reasons we think that might be occurring, and so this is the difference in strobe resistance that we saw this year. So if you look at our end of the year testing, we typically see high prevalence of strobilurin resistance in Dr. Secor's fungicide testing. However, in the spring, we would see significantly lower levels of that. And this year, we detected a lot more resistance early than we would have thought. A potential explanation for that is what you see here on the left side of the screen, where Dr. Secor and his technician Viviana tested the germination rate at different temperatures for resistant and sensitive isolates. So what you see is that Cercospora isolates that are sensitive to strobilurins have a higher germination rate at lower temperatures. And because of that, or actually the better way to frame that is that resistant isolates need higher temperatures to start germinating and infecting. And so this year, with the atypically warm winter and leading into a pretty early spring where we had warm temperatures, you had those resistant isolates emerge earlier than they have in the past. But there's a really delicate interpretation of that result that I want to make sure we're really clear about, and this will be a theme throughout this talk. Even though we detected really high levels of strobe resistance on a per sample basis, the total DNA content within each of those samples on average was about 63% resistant. So really you're wavering really close to that 50% line on the quantity of resistant and sensitive infection that's occurring in your fields. So there's a big difference between a per sample or per field metric of fungicide resistance and then boiling down to the actual individuals contributing to that and how many of those are present. To cover real quickly, we saw very little resistance to DMIs earlier and very little resistance to Topsin in those early latent detection samples. Now, why do we care about detecting latent infection? And Eric did a nice job. Eric and Ashok have done really nice jobs of highlighting this in their trials. But what we see if we look at field data collected and shared with us by American Crystal Sugar across all of their acreage, if you take each of the fields and bin them into categories based on when they started their Cercospora fungicide management, we see that fields that started managing for Cercospora in that last week of June had the highest recoverable sucrose on average across the last 7 years. So we haven't put the 2024 data in here yet. This is 2017 to 2023. And if you can see the color differences here, and I apologize for this because there's not that many colors we can use, but if we want to look at a low CLS pressure year and we zoom into, say, 2022, so 2022, fairly low CLS pressure year, and you can see on that purple line as you move across the top here that there isn't a big penalty in recoverable sucrose per acre detected here if you were starting fungicide applications later in the year. However, on a high pressure year like 2020, when there were a lot of local epidemics of CLS, that actually starts a steep decline immediately as you step into the first week of July. So you're seeing about a 5% reduction in your recoverable sucrose per acre if you're missing that initial timing. And I show this just to really highlight the critical importance of that first application's timing to get ahead of this disease so that you're not fighting that exponential curve. One other thing I want to highlight too, and this is a study that we've done for the last 3 years now. I'm only showing data from 2022 and 2023, essentially because we're still working on sorting through the data. These 2 years we had really consistent fungicide applications to compare. When we've done this sort of field sampling throughout the season, the data gets a little messy when you're looking at different number of fungicide applications depending on the year you're looking at. But what we did in this study was we selected fields and had co-op agriculturalists go out and collect samples from those fields before each fungicide application and then following each fungicide application but before the next one, so that you could track the resistance levels in response to fungicide applications. And one of the interesting things that happened in these fields that received 3 fungicide applications through Crystal Sugar's growing region is that we saw our strobe resistance frequency going into the first fungicide application was on average 50% of the isolates in that field that were sampled, and that dropped significantly after the first DMI EVDC application for those fields. You get a slight rise as the year progresses on, and following a tin topsin application, we saw no decline in the strobilurin resistance levels. However, there's a big spike that occurs after a final application that included a strobilurin, which makes sense because you're effectively killing off everything that couldn't survive a strobe application, which just leaves highly resistant isolates, and that's why we see a big spike in strobe resistance at the end of the year in these fields. But I show this again just to highlight that the fungicide resistance metrics that we have are very dependent on when we're sampling throughout the year. And your management practices are pushing this population of Cercospora in whatever direction it needs to try to circumvent, to try to circumvent those management practices and sort of outsmart our fungicide applications. Now, we've been doing a lot of testing for cross resistance between different fungicide chemistries in my lab with single individual isolates. So one individual, how capable is a single individual at being resistant to multiple chemistries? And what the figure you're seeing here is a correlation matrix where values closer to 1 would indicate complete correlation between resistance, and values closer to 0 would indicate no correlation. And to highlight that, you can see here that like a 1, when you're comparing the same compound, shows up as perfect correlation, which makes sense. And when you start comparing different compounds, you get significantly lower correlation coefficients. And you can think about this in loose terms as saying you have 100% correlation between headline and headline. That makes sense. 18% correlation between topsin and headline. 40% correlation between headline and tin. And to highlight how this data is useful for us, this is a great argument for tank mix partners. Especially so when you look at topsin and tin, there's a very low correlation coefficient here between those two compounds, meaning that isolates have a hard time, so Cercospora has a hard time at being resistant to both of those chemistries. So when applied in a tank mix combination, if tin is getting half of the isolates in that field, topsin is getting the other half. And as you can see, there's a few high correlation coefficients when you get within the triazoles or the DMIs. So Proline and Domark have a really high correlation coefficient. And in our lab results, we see that isolates that are resistant to one are typically resistant to the other as well. But that's fairly leaky when you get to other fungicides such as Inspire. You have a lower correlation coefficient there. And the same goes for Provosol, but I don't have that data up here at the moment. So having now covered the fungicide data from the latent study and our cross-resistance data, I want to jump into some population dynamics. So hopefully you had a good bit of coffee today, because this is where the water gets deep. So a project that we've had in the Red River Valley in collaboration with a large group of scientists we've been sequencing whole genomes of Cercospora isolates that have been collected every year. And we're slowly working our way backwards to the earliest collected samples from 1997 by Dr. Secor. In the meantime, though, we've sequenced populations from 2016, 2017, 2021, 2023. We plan to sequence population from 2024. And the way we use this data is to look at population dynamics or changes in population structure over time that we can then try to relate to any changes in management practices. And a good way to visualize that is with what's called a principal component plot. And The next few slides where you see these principal component plots with lots of little tiny data points on there, the important thing you want to focus on is the general shape of the clusters here. And when we look at that clustering data, if you look at population 1 and population 2 here, the complete separation of those populations in this plot indicates that those are genetically distinct populations. And if you look at the comparison between population 1 and population 3, what we would interpret in this case is that you have population 3 branching off of population 1. So this is a good way that we can look at whether or not we have a diverging population or separate populations, and then compare management practices and metadata around those collections to see if there's anything that correlates with these events. And one of the ways we've done that is to establish a baseline over the last few years of what the population structure should look like. So this is 2016 isolates. Again, important just to pay attention to the overall structure of the population, so the big oval here. And then as we page through time here, you'll see in 2017 we have roughly the same population structure, approximately the same shape. In 2021, we get a different shape. And what you see branching off on the right side of the screen is a divergent set of Cercospora isolates. Now, this was interesting to us because this was the first year that CR Plus was introduced, and being a great source of Cercospora resistance, it makes sense that this would be a fairly strong selection pressure and drive the population in one direction in order for it to try to adapt. To confirm that, we sequenced a population from 2023, and essentially with the now all the isolates shown in pink here, we're able to confirm that that specific divergence does appear to be related to CR+. And this cluster on the far right, over very far right here, are actually exclusively isolates derived from CR+ fields. Now, how we leverage this data to get to the point where we can actually create a rapid marker to screen for this is by looking at all the different mutations that are associated with that trait. So every dot you see on this plot here is a separate mutation. There's over 1 million of them. And we run essentially an association mapping analysis in order to figure out if there's regions of the genome that are specifically associated with this trait. Then we zoom in to see if we can identify the genes that are there. And long story short, we have these 3 interesting regions where you have divergence and high indicators of adaptation on CR+, and this is what we're working on investigating right now. Already, Dr. Bolton and I have a student who has knocked out a gene in this first region. Sorry, I'm gonna go back a little bit. Knocked out a gene in this first region in an isolate collected in 2009. And that isolate, when we take that gene away, becomes very aggressive on CR+ lines. And so we're very confident now that we have a good gene and a good genetic marker for CR+ adaptation. However, what you can also see is that there's other loci here, there's other parts of the genome and other genes that are going to be involved in this. And so this is, as a geneticist, exciting work because you have this sort of additive, adaptive environment, and we're really interested in investigating how that adaptation occurs. As a grower and production person, scary because you have adaptation and now optimization occurring at other loci to essentially have the pathogen compare what genes are the most optimal in combination to be successful in the face of the management practices we put out there. Now, we use this data in a couple other ways as well. So tin resistance has always been a sort of elusive trait to get a good genetic marker for. And using this whole genome sequencing data, we've also been able to identify some significant associations related to tin resistance. And those are the highlighted green mutations here on this plot. One of the cool parts about that is when we zoomed into what kind of gene might be there, we found a glutathione transferase gene, which I actually didn't know what those were until I started reading into it quite heavily. They're commonly involved in fungicide resistance and herbicide resistance, actually. But when we zoomed in on that gene, it became extremely interesting to us because there's a substrate binding pocket that looks like tin would fit in it, and almost all of the mutations, which you can see in the pink gene structure here, all the little hash marks at the end of that on the left side, are mutations that occur in the binding pocket for this particular enzyme. And in our populations, it's really only present in two forms, and one form, when we look at isolates with the first form, are fairly resistant to tin. The other form are sensitive to tin. And so again, we've been able to develop and are working on validating a marker for tin resistance that we can start running for rapid screening. The last bit of data that we're using, or that we're extracting from this whole genome sequencing data, is to look at the frequency of these mutations across the populations. And this is where I think it's important, again, to reiterate that when we do fungicide resistance screening, we do it in two different ways. We do poison plate assays on individual isolates, so we get a per individual score, and that's oftentimes an EC50 value. And then we also do population level screening, where we take a whole bunch of isolates from a single field, pool them, and then see how many of those isolates are capable of being resistant to any particular fungicide. And so historically, so in 2021 and 22, there were really high levels of tin resistance detected on a field-by-field basis. However, when we zoom into the individuals in that field, typically right around 30% of those individuals are resistant. So though we have widespread resistance, it's sort of patchy in a field, if that makes sense. And when we zoom in on our sequencing data that comes from those end-of-the-year collections, we can look at the mutations that are related to headline triazole resistance, topsin resistance, or our tin resistance now. And in 2016, 2017, 2021, and 2023, we see that the range of those isolates at the end of the year, as far as the prevalence for those mutations, is fairly consistent. You have higher levels of resistance to some of the DMIs that are more commonly used multiple times in the year, fairly low to moderate levels or percentages of resistance to strobilurins and to topsin, depending on the year. And again, that tin resistance on a per individual basis sits right around that 30% mark on average. And so the reason I really want to highlight this is there's a lot of surveys that get done on headline resistance, on topsin resistance, that show really high prevalence of topsin resistance on a field-by-field basis. But similar to our cross-resistance data, when you get to the actual individuals in that field, most of those individuals struggle to be resistant to more than one or two chemistries. And that's why tank mixing is a very useful tool for fungicide applications, and specifically rotating through as many modes of action as you can in a given year. So some ongoing research that we're currently finalizing. Dr. Secor and I, our expanded CLS risk model to aid in management decisions. We've got gene knockouts generated for our CR plus adaptation traits and our tin resistance traits, and are working on validating those phenotypes in the lab. That's now led to us having PCR-based marker assays for those traits so that we could rapidly detect those, similar to how we detect mutations for the latent infection study. We're going to continue our whole genome sequencing effort, specifically focused on 2024, especially, which I think is important given the increased disease pressure we saw on CR plus varieties. It would be really good confirmation of that trend we saw of the population diverging. And then we are actively working to sequence backwards to the 1997 population that Dr. Secor has. And that's going to be important because if we get sequenced populations from each of those years and we cover the time span of some of these changes in management practices, such as the introduction of triazoles and strobilurin fungicides in the early 2000s, or when tank mixing became exceptionally popular, we should be able to then use that data, look at how long it took for adaptation to any one trait or combinations of traits to happen, and then be able to forward project how long any new management practice might be durable for. So whether that's looking at how durable CR plus is, and when that fully breaks down, you have now a time span to look at the next resistance that gets introduced, and then in combination with the CR plus resistance, and then the same could be said as, same could be compared and forward predicted for any new resistance trait paired with any new fungicide chemistry that comes out. So hopefully that will be extremely useful data for us to model the durability of management practices moving into the future. Tons of acknowledgments to be had here. especially Dr. Bolton's lab at the USDA, Dr. Secor's lab at NUSU Plant Pathology, who contributed to this work, and then all of the co-op individuals who we work with and the agriculturalist staff who submit all these samples. This year we're asking for less samples from the agricultural staff, so I hope that's a good thing. We're targeting our latent infection to just a couple weeks instead of trying to capture a month and a half of data. But yeah, I can take any questions you might have. Fire away.
When do you expect the 24 results, or when will you have them done? So will we in time for our Scottsboro spraying?
Yeah, so we have the isolates growing right now, so we should be able to extract, oh, sorry. So the question was, with our 2024 whole genome sequencing, what's sort of the expected timeline? Ideally, like a perfect timeline, Dr. Secor, or moving into the future if Dr. Secor retires, through my lab, when those isolates come in for fungicide testing at the end of the year, we'll immediately port those to a sequencing pipeline, and we can have those before the first of the year. This year, we got the isolates just a couple weeks ago from Dr. Secor. We have now fungal tissue. We'll extract DNA over the course of a couple weeks, and so within the next few months, we'll have that 2024 population sequence. But we can realistically have a fairly quick turnaround time on that population tracking throughout the years. Fire away.
Have you made any progress on cross-resistance testing on the other triazoles, TopGuard enabled, to try to get a better handle on where they're going to fit?
Yes. So, well, and I shouldn't say yes for myself, but Dr. Secor did some testing with flutriafol, which I believe is TopGuard, but I need my cheat sheet here. That is cool. So, and I should repeat the question. The question was, have we made any progress on testing other triazoles outside of the 4 that have been sort of the hallmarks of the testing here? So we've done 2 things. Dr. Secor's incorporated some other DMIs into his testing so that we can put these in either the category of being cross-resistant with Proline and Domark, or cross-resistant with Inspire and Provosol. So, for example, TopGuard ended up being cross-resistant with Proline and and Domark. We've also sort of gone back through Dr. Secor's historical data where he tested, I think there's 9 total triazoles that are in there, and we're working on putting those into a cheat sheet to send out for cross-resistance. Any other questions? All right, I will be around as long as you need me today, so